Proteins take on an enormous variety of shapes, stabilized by several types of bonds. But how does each protein get there? This is the realm of protein folding, and how it happens is amazing. It's almost as if identical strips of paper, left to themselves, spontaneously fold into intricate origami cranes. Not once, but almost every single time, while other strips of paper somehow almost always fold themselves into origami butterflies. Of all the shapes paper could be folded into, how does one so consistently fold into a crane, while another so consistently makes a butterfly? Adding to this challenge is the speed of the process. Proteins fold mind-bogglingly fast, sometimes on the order of milliseconds. It's too fast for the molecule to do this by a random process. So how does this happen? In this lecture, we'll go through what we know and some of what we don't understand about the fascinating details of protein folding. I'll also describe some deadly consequences that result when proteins do not fold properly, which includes many degenerative conditions such as Alzheimer's, Parkinson's, and the so-called Prion Diseases. I'll also talk about the convention defiers, proteins that don't fold into a distinct 3D structure at all, and yet get away with it without causing us harm. From the last lecture on protein structure, you may have had the impression that protein folding would be a simple function of geometry of the amino acids involved. After all, they each have fixed dimensions and properties, and the resulting second-order structure can be predicted with a fair degree of accuracy. But the more we've studied protein folding, the more evident it's become that folding is anything but simple. Some illustrations you may come across are, to be blunt, only a guess about how the process is occurring. Nonetheless, advances are being made. So far, simulations can approximate the folding of a few small-sized proteins. You may have heard of Folding at Home, a distributed computing initiative to allow anyone to volunteer unused processor time on their computers to help simulate folding of specific proteins. However, while we're pretty good at predicting regions of a protein with alpha helices and beta strands, we're still far from a general algorithm capable of using primary structure to predict how secondary structures are arranged into that folded 3D conformation. We'll examine some of the challenges that make this such a difficult problem to solve. But let's start with what we do know. In the 1950s, the first experiments began by taking a protein out of the cell, unfolding it, and then seeing if it could refold in a test tube, independent of any cellular factors. The protein Christian Onfenson picked was the enzyme ribonuclease A, also known as RNase, which turned out to be a serendipitous choice. RNase is relatively small as proteins go, about 100 amino acids. It is also extraordinarily stable. While most enzymes are exquisitely sensitive to changes in temperature and pH, ribonuclease A is not easily denatured, thanks to disulfide bonds connecting cysteine molecules in 4 different locations. To completely unfold RNase, you must break the disulfide bonds using a chemical agent such as mercaptoethanol. You can break the hydrogen bonds using something like urea. Use both together and that completely unfolds RNase, which is then no longer capable of enzymatic activity. Once the enzyme is unfolded, can it refold outside the cell? The answer turns out to be yes, at least partly, as Onfenson showed in work that won the 1972 Nobel Prize. We call this renaturation because the protein has been returned to its native or natural state. But the renaturation was not perfect. Sometimes cysteines misaligned and formed the wrong disulfide bonds. Nevertheless, the fact that at least some renaturation occurred properly meant that the folding was dictated by the sequence and not by some cellular factor. Experimentally altering amino acid sequences backs this up. Changing amino acid sequence changes 3D structure. Still, Onfenson's choice of RNase was fortunate, and here's why. Not many proteins are as close to self-sufficient about folding themselves as ribonuclease A. Many proteins have little or no ability to refold after you denature them. Cooking an egg provides a good example. Over the course of a few minutes, the liquid egg begins to solidify, and when you're done, it's completely solid, albeit fluffy. What happened? Egg proteins, before you cook them, are dissolved in water, where they are folded so that any amino acids with hydrophobic, water-fearing side chains are on the inside of the protein. Hydrophilic, water-loving amino acids are on the outside, facing water. Adding heat with cooking breaks hydrogen bonds, and all non-covalent forces stabilizing the folded structures so that the proteins in the egg begin to unfold or denature. But if you unfold hydrophobic side chains in an aqueous environment, they're going to do something else to avoid water. They invert and associate with each other in intermolecular interactions, making large aggregates. Meanwhile, what happened to the hydrophilic amino acids? They're left to move to the inside. And what do they take with them? Water. By the way, denaturing proteins in harmful bacteria occurs as you cook anything, killing them, which is why you cook in the first place. All that rearranging of egg proteins is not reversible. And just as you can't unscramble egg protein that has been denatured by heat in a pan, you would have a hard time renaturing many proteins after you unfolded them. Some proteins begin folding as they're being made, meaning that the beginning of a polypeptide chain can start folding before the rest of the sequence is attached. This simplifies the number of available options. For example, if side chains of amino acids in the first few amino acids have already folded when interacting with each other, they are less likely to be available to interact with amino acids added later, thus reducing the likelihood of interactions producing incorrect folds. By contrast, a protein that is being cooked or denatured doesn't have such restrictions. First, many other random motions of amino acids in the polypeptide become possible, enabling interactions between regions that normally wouldn't associate. The result? An improperly folded protein that's inactive. Second, amino acids of one molecule of a polypeptide can also gain opportunities to interact with those of another polypeptide, forming a protein aggregate like we saw in our cooked egg. And there are so many ways to fold, at least in principle. A quick back-of-the-envelope calculation by Cyrus Leventhal in the 1960s showed that the number of ways a small polypeptide of just 100 amino acids could fold would be in the same ballpark as the number of atoms in the universe. Fortunately, there are a few simplifications resulting from structure. The double bond holding the oxygen atom to the carbon in the peptide bond is electron-rich and can electronically rearrange into an alternative structure that turns the adjacent carbon-nitrogen bond into a double bond. Both of these structures exist interchangeably through a phenomenon called resonance. Any resonant double bond is fundamentally different from single bonds as it is locked in place and cannot rotate, whereas single bonds can. Because of this, we can assume the peptide bond is fixed and cannot rotate. The two single bonds on the atoms on either side of it, though, can completely freely rotate. This limits the polypeptide to conformations resulting from rotations around those two single bonds, the nitrogen-carbon alpha bond called phi, and the carbon-alpha-carbon bond called psi. Most theoretically possible phi and psi angles give rise to structures that are unstable because such angles would result in putting two atoms too close together. We can get a good overview of the rotational angle parameters using what's called a Ramachandran plot, which shows all theoretically possible rotational angles for phi and psi. In fact, the phi and psi angles that do give stable structures correspond mostly to rotational angles found in well-known secondary structures. Notice that alpha helix clusters at negative 45 degrees for the phi angle and for the psi angle. Bond angles that give rise to beta sheets and to the wider helix of collagen are clustered in the upper left region of the chart. And that's about it. The other half of the graph is almost empty, except for the otherwise rare bond angles adopted by left-handed alpha helices. Another consideration for folding is in terms of energy. We know experimental evidence that protein folding is thermodynamically favorable, whether by releasing heat, increasing entropy, or both. One way to envision thermodynamically favorable processes is to think of them as downhill reactions, just as a ball rolls spontaneously down a slope. Formation of bonds or forces that are thermodynamically favorable includes several known to stabilize protein structure. They include hydrogen bonds, ionic bonds, hydrophobic effects, and disulfide bonds. Researchers think folding proceeds energetically by an overall energy funnel mechanism, not unlike that of a skier moving down a long hill with numerous mini hills along the way. The unfolded protein is at the top of the hill in the highest energy state. As the protein makes its way down the energy hill, it accumulates favorable interactions that lower its energy progressively. Just as different skiers could take slightly different paths down to the bottom of the hill, not every molecule of a protein must take an identical path to the bottom of the energy funnel. The path is not always smooth either. Along the trail to the folded state, a protein might fall into a local energy crevasse and would need an energy barrier to overcome as a result of a condition described fittingly as frustration. But overall, the native conformation is the lowest energy state that the protein can achieve. And as folding proceeds, interactions that favor the native state accumulate. Each favorable interaction lowers the energy state while also narrowing the range of remaining options for the folding of the polypeptide, in effect, increasing the odds of the protein achieving its native form. But that, it turns out, isn't the whole story. Remember that even small proteins, 100 amino acids or fewer, like RNase, are taken out of the cellular environment, can have delays or small errors when they refold. And some larger proteins, once unfolded outside the cell, fail to regain their native conformation at all. Clearly, there is something different about the cellular environment that allows them to fold correctly. But conditions within the cell are actually, in some ways, less favorable for proper folding of proteins because the inside of the cell is very crowded. A protein trying to fold in this environment would be in somewhat of the same situation as a person trying to strike a yoga pose in a tightly packed subway car. Other occupants of the space would restrict movement, and their limbs might get entangled with those of the would-be yoga practitioner. In the cell, this kind of crowding tends to bump partly folded proteins up against each other, increasing the likelihood of misfolding or aggregating into mistaken clumps. This is where helper proteins come in. To assist in correct protein folding in these challenging conditions, all cells have helpers called molecular chaperones. Somewhat like their human-sized counterparts, molecular chaperones work, in part, by preventing inappropriate interactions from folding, if you know what I mean. The term molecular chaperone is used to describe any protein that helps another protein to achieve its functionally active conformation, but it does not remain associated with the protein once folding has taken place. There are several classes of chaperones that work together to ensure proteins are properly folded. Some help in the actual folding process, while others work with misfolded proteins to allow them to refold correctly. Other chaperones tag hopelessly misfolded proteins for destruction. Chaperone proteins were originally identified in cells transiently exposed to high but non-lethal temperatures. Heat, as you know, can have the effect of breaking weak interactions like hydrogen bonds and could result in partly unfolding cellular proteins. The response of cells to heat is to make large amounts of proteins named heat shock proteins. We know now that these heat shock proteins, or HSPs, are molecular chaperones that help refold the partially heat-denatured proteins in these cells. In fact, under any stressful conditions with an increase in misfolded or unfolded proteins, cells increase their production of molecular chaperones. But all cells make chaperones even when they are not under such stress. That's because when polypeptides are being made, their hydrophobic regions are at risk of associating with hydrophobic components of another partly folded protein molecule in the crowded environment of the cell. This could lead to formation of toxic, misfolded aggregates. Two types of chaperones prevent this from happening. One binds to the hydrophobic regions of a nascent polypeptide and blocks interactions with other proteins. As the polypeptide grows and the hydrophobic regions avoid any incorrect interaction, the chaperone dissociates, allowing for proper folding. A second class of chaperones, called Chaperonins, prevent harmful folds by providing a sort of secluded space away from the other molecules in the cell, where a protein can fold undisturbed without making contacts that would lead to misfolding or aggregation. Chaperonins have a structure that is reminiscent of a bin with a lid. The inside of the bin is where the magic occurs. When the protein enters the bin, amino acids with hydrophobic side chains extend from the walls of the bin. When the lid goes on, a conformation change in the bin occurs. Hydrophobic side chains invert, and hydrophilic side chains are exposed and appear on the walls of the bin. When exposed to the hydrophilic side chains, the hydrophobic side chains of the enclosed protein align with each other to avoid the hydrophilics. And this is the start of what would happen if the protein folded by itself in an aqueous environment. The difference here is there are no hydrophobic side chains available from other proteins, so there's no opportunity for intermolecular interactions to form aggregates with other proteins. Thus, the chaperonin favors intramolecular folding and blocks intermolecular aggregation. But all proteins eventually need to be replaced. So there are chaperones that identify and target such proteins for degradation. HSP70 is a chaperone in humans and all other eukaryotic cells, and it identifies misfolded proteins for recycling. It binds to exposed hydrophobic regions on the surface of misfolded proteins and attracts an enzyme that covalently links a small protein known as ubiquitin to the exposed region. The ubiquitin protein acts as a tag, marking the misfolded protein for destruction, which takes place in a cellular recycling center called the proteasome. In the proteasome, the ubiquitin tag is released to bond with another target, while the defective protein is broken down into smaller pieces with the help of other enzymes known as proteases. The proteasome releases these short peptide pieces, which later are further dismantled by cells into their constituent amino acids. Basically, we have protein enzymes replacing other protein enzymes. Whene'er my proteins go kaput, if they are past their prime, the cells will act to soon replace all of my old enzymes. They know which ones to break apart. Ubiquitin's the sign, a marker for proteasomes to find the old enzymes. So far, so good. But some of the most mysterious diseases result from protein folding gone wrong. These are the so-called Prion diseases, also known as transmissible spongiform encephalopathies, or TSEs. Prion diseases affect humans and other animals. They are a group of degenerative disorders that affect the brain, creating microscopic holes that make the brain look like a sponge. Mad cow disease, or bovine spongiform encephalopathy, BSE, is one of the best-known prion diseases. Researchers first began noticing mad cow disease in the 1980s based on its similarity to another disease in sheep known as scrapie. Animals suffering from these diseases exhibit behaviors consistent with neurological damage. Tracking the molecular roots of the disease was at first maddening. The diseases were infectious, but they appeared to violate all known rules for an infectious disease. Researchers rounding up the usual suspects could not find any known causative agent, whether a bacterium or a virus, that might be responsible for spreading the infection. Stanley Prusiner at the University of California at San Francisco ultimately identified the infectious agent as a protein, a proteinaceous infectious particle he called a prion. The protein that prions are made from was named PRP, standing for prion protein. Yeah, it's redundant, okay. A prion is a protein, kind of like saying it's a PIN number. But after the fact, it's a little hard to change that. That a protein could be infectious by itself was unheard of at the time, and PRP turned out to be a cellular protein found on the membrane of healthy cells, though its function to this day remains uncertain. Not only was an infectious protein unexpected, its mode of action was also a complete surprise. PRP exists in two forms. In a healthy organism, only one form, known as PRP-C, is found. It contains mostly alpha helices. In infected organisms, a second form of PRP is also found, known as PRP-SC. The SC stands for scrapie. The second form has aberrant secondary and tertiary structures, consisting mostly of beta sheets rather than primarily alpha helices. In the brains of prion-infected organisms, the unwanted beta sheets of the abnormal prion protein clump together to form aggregates. These aggregates take the form of long, highly packed beta sheet structures that are not soluble in water and therefore form very stable structures. Known as amyloid structures, these tightly packed beta fibrils are in contrast to the water-soluble beta sheets common to many proteins. It is believed that the misfolded PRP-SC proteins have lots of hydrophobic side chains that self-assemble to make these insoluble fibrils, which quickly lead to cell death. So where did the misfolded beta sheet fibrils come from? Bizarrely, not from the primary sequence of amino acids. Instead, the strange fact emerged that misfolded PRP-SC proteins combine to normal PRP-C proteins and somehow convert the normal alpha helices into an infectious beta sheet fibril and continue the cycle. So prion diseases are caused by a misfolded protein that somehow causes its normal counterparts to fold in abnormal ways and remake themselves in the same misfolded image. All known prion diseases in mammals affect and destroy brain or neural tissue. All are progressive, meaning they worsen and spread. There is no known treatment as well, and all are fatal. Prions are thought to be the cause of several human diseases, including the type of dementia called Creutzfeldt-Jakob disease, or CJD. Fatal familial insomnia and Kuru are also human diseases caused by prions. We don't understand exactly how prions are transmitted from one individual to another. Prion diseases sometimes appear without a known cause. Sometimes they're inherited, sometimes they're acquired through exposure to infected material. For example, it has been suggested that the appearance of a variant of Creutzfeldt-Jakob disease in Europe may be linked to consumption of prion-contaminated beef from animals that had mad cow disease. Like prion diseases, Alzheimer's and Parkinson's are also associated with misfolded proteins that aggregate. In the case of Alzheimer's, tangles made up of fragments of misfolded proteins are seen outside of nerve cells, forming visible plaques called beta amyloids. In addition, a protein called tau can go wrong and form deposits within the neurons. It isn't clear whether one of these or both are responsible for the damage to neurons that causes cognitive decline. In Parkinson's, clumps of a small misfolded protein called alpha-synuclein form within neurons in the region of the brainstem known as the substantia nigra. The formation of these clumps, called Lewy bodies, is associated with death of neurons that produce dopamine. Lack of dopamine, a transmitter used in neural signaling, leads to symptoms of Parkinson's. So given the many debilitating diseases that seem to result from misfolding of proteins, researchers would love to reverse or halt protein misfolding. If I should ever chance to be on a protein making up a prion, I'd twist it and, for goodness sakes, stop it from making fold mistakes. Imagine identifying small hydrophobic molecules that can enter the affected cells and specifically bind to the misfolded proteins, preventing their aggregation. Imagine making a chaperone that could even promote refolding back into the native form. Drug research began around 2003 to develop pharmacological chaperones known as Pharmacoporons. Some studies in cell cultures and animal models suggest that this approach could be useful in treating or even preventing a variety of protein misfolding diseases. Up to this point, we've been focused on the multiple mechanisms that cells have to help fold proteins correctly, refold those that don't, and dispose of the worst cases, plus efforts to avoid the devastating problems that can arise from misfolding. So our final topic in this lecture may come as a total surprise: the existence of proteins that are described as intrinsically disordered. These intrinsically disordered proteins, or IDPs, as their name implies, have no fixed 3D conformation in at least part of the protein. And yet, surprisingly, despite the name, this is not a harmful aberration. Numerous proteins, perhaps 40% of all proteins, may have at least one region that is intrinsically disordered. The big question is, if structure determines function, why would so many proteins naturally have portions with no fixed shape? Well, confusing as it might sound, their lack of fixed structure makes possible a different kind of function. Yeah, I know, this flies in the face of what I've been talking about so far in this lecture. It's also in conflict with a message beginning biochemistry students often get about enzymes, which states that an enzyme is like a lock and the molecule it acts on is like a key that fits into the lock. This simple perspective implies that proteins are fixed, rigid structures with precise X, Y, and Z coordinates for every atom. Well, that turns out to be an oversimplification. Proteins are not rigid structures, but rather flexible. We can think of their coordinates as average values and their structures as having a bit of give and take. Sort of like a memory foam mattress can adapt to someone sleeping on it, but springs back to its original structure when the person leaves. Intrinsically disordered proteins take this flexibility to the extreme. You can think of intrinsically disordered as meaning fluid. Some have no fixed shape anywhere throughout their structure and are called fully disordered, such as cyclin-dependent kinase P21. Others are partly folded into a fixed shape, but also have regions without a fixed shape. Such proteins are described as partly disordered. An important tumor suppressor protein, such as P53 or BRCA1, has significant portions of its structure that are intrinsically disordered, and that lack of fixed structure allows it to interact with a wider variety of molecules. A protein with a fixed 3D conformation is limited in the molecules it can interact with. By contrast, an IDP could bind to a wider variety of partners, adopting a different structure complementary to each partner, like a memory foam mattress. Indeed, intrinsically disordered regions are found in proteins that are known to interact with multiple molecular partners, with functions that include signaling and regulation. During the 21st century, IDPs have come to be recognized as a class of proteins in their own right. Analysis of the sequence of proteins known to be disordered reveals that they are unusually rich in hydrophilic amino acids, such as serine and cysteine. And with that, it brings us full circle to one of the forces favoring protein folding. the association of hydrophobic amino acids inside of a protein to avoid water. That means primarily the branched chain amino acids such as leucine plus phenylalanine and tryptophan. Conversely, a lack of those hydrophobic amino acids in IDPs may lead to and determine their disordered structure because there will be much less of the hydrophobic effects that drive the folding process. So inherently disordered proteins have vastly expanded our ideas about protein structure and folding. For some proteins, function does result from proper folding into a regular 3D structure, as students have been taught for decades. But for other proteins, function may arise from exactly the opposite: the inability of a protein or a part thereof to form a precise structure. This may be what allows for flexible interactions with a much wider variety of other biomolecules. So let's wrap this up. The take-home message I hope you remember about protein folding is this: how or whether a protein folds is determined by two things: the primary structure comprised of amino acid sequence, and a thermodynamic energy funnel that goes downhill. For some proteins, chaperone helpers that facilitate folding are essential. Another point should also be clear: interfering with a protein's ability to fold properly can have severe consequences for human and animal health. In the next lecture, we'll learn how a carefully folded protein like hemoglobin makes use of flexibility in responding to the body's changing needs for oxygen. It's like an origami butterfly that flutters.